Hello everyone, myself Krushna Pavan and welcome to my poster multiHIVE. It's a hierarchical multimodal deep generative modeling for single cell multiomics and I'm a computer science student at IIT Kanpur. Let's begin with a bit of background. With the advancements of technologies, now we are able to extract both RNA sequencing data and surface protein data from a single cell simultaneously using which using this technology we were able to extract both RNA sequencing and protein expression from a single cell. Now our task here is to combine these two different modalities and have a unified view to gain more insights into the data. This technology is called site-seq technology. Now let us have a quick look into the methods section. As name suggests, multiHIVE consists of a hierarchical VAE which consists of two private encoders to encode different modalities like RNA and protein in their latent space. And then we have a shared encoder which takes both RNA and protein expression and embeds them into latent space in an hierarchical model. And finally, the shared latent representation along with the private latent representation goes into respective decoders to generate back the gene expression and protein expression, and the whole model is trained using elbow loss. Moving further, we have rated our model with different tasks with you can see which are site-seq integration, integration with RNA-seq, protein imputation and biological capture. And here are the list of data sets over which we have performed this different task, and you can also see different metrics that are used to evaluate different tasks, and we finally benchmark these different tasks over 4 different methods which are MOFA, Seurat, multiHIVE and total VI. Let us have a look into the quick results of integration of site-seq data using PBMC. The first figure, each dot indicates a cell of cell with a similar cell type. We can see that all the cells are integrated, all the cells with same color are together, which indicates that the integration is happening. We can also see quantitatively. Uh, Now, quantitatively, what we have done is we have used latent clustering at different resolution and compared it using the ground truth to get the NMI values across all the different methods. We can see that multiHIVE was able to perform best in comparison to all other methods. Next, to increase the complexity in the data integration task, we have created a simulated data using the PBMC Stephenson data. We can see that again multi-hives embedding where each color represents a cell type and each dot represents a single cell. We can see that all similar cell types, similar color is together, which indicates the integration is happening qualitatively, and we can see that quantitatively also using NMI and ARI values, multi-hive was able to perform best, whereas the MOFA was not able to integrate this dataset well. And next, the result is on protein imputation. For this task, what we have done is we have removed few of the proteins from the data and tried to predict those proteins and compared it against the ground truth proteins. We see that the protein expression, the Pearson's correlation of the protein expression is highest for our multi-hive model, and the RMSE error, that is root mean square error, is lowest for our multi-hive model. Here also a glimpse of proteins and their R values in comparison to ground truth. Next, we have the results of breast cancer. Here, to see what insights are being captured by each of the lower dimension embeddings, we have passed each of the lower dimension embeddings, which is Z1 and Z2, through the decoder and taken the regenerated RNA expression and deconvoluted using proteins, deconvoluted it using programs and CNMF into programs using consensus non-matrix factorization, which is called CNMF. we can see that the Z1 is able to capture the programs uh, which basically captures the cell types. Like the program 1 is basically trying to capture the two cell types, which are T and B cells, and so on. And then further we can see that Z2 was able to capture pathways which are more generic across the cell types, like from T cell activation to the cell cycle process, from program 1 to program 7. Next, we have looked into thymocytes. Thymocyte development, it's a continuous development process data set. Again, we have done the similar thing to this data set, and we looked into the pathways of Z1. We can see the pathways, meaningful pathways for these particular Z1 deconvoluted RNA expression. And we also looked into few of the gene expressions over the pseudotime of this data. We can see the results which are aligning with the background knowledge. And here is the highlight. MultiHIVE can do multiple tasks like site integration, protein imputation, integration with RNA-seq. It is able to capture at and cross cell type levels data, and it's a promising tool for decoding cellular systems and gaining insights. And the code is available at this particular link. Thanking you, myself once again, Krushna Pavan, and you can contact me over this particular mail ID. Thank you.